In this section, we will talk about the mechanisms by which chemical messengers act in types of cell receptors. As we discussed earlier, that the interaction of ligand with receptor is just the beginning in the cell signaling pathway. For the cell to communicate effectively, it demands a diverse collection of receptor-mediated pathways that execute their unique functions. If you recall what we discussed in the previous section, the second stage of cell signaling pathway was transduction, and there was a mention of series of events. Those series of events can be divided into 3 categories: ion channel activation, G protein activation, activation of enzyme activity within the cell. But these activations can only take place after a chemical messenger binds with a receptor. There are many types of receptors in a single cell. We have already talked about the internal and external receptors briefly. Let's talk about them in some detail now. Starting with intracellular receptors. Intracellular receptors respond to small hydrophobic ligands that can travel across the plasma membrane. As you can see here, the ligand passes through the cell membrane and binds to the intracellular receptor. Once inside the cell, many of these molecules bind to proteins that act as regulators of mRNA synthesis. That is, transcription to mediate gene expression. If we talk about the cell surface receptors are membrane-anchored proteins that bind to ligands on the surface of the cell. Ligands that interact with cell surface receptors do not have to enter the cell. So many kinds of molecules may act as ligands. The cell surface receptor has 3 main components: an external ligand binding domain, a hydrophobic membrane spanning region, an intracellular domain inside the cell. There are many kinds of cell surface receptors, but here we'll discuss 3 common types and relate them to the activation mechanism that we mentioned before. Ligand-gated ion channels, G protein-coupled receptors, receptor tyrosine kinases. Ligand-gated ion channels. The name suggests their function. Ion channels that open when a ligand binds to the receptor. These allow specific ions to pass through the membrane. When a ligand binds to the extracellular region of the channel, there is a conformational change in the protein structure that allows ions such as sodium, calcium, magnesium, and hydrogen to pass through. In our video on conduction of action potential in heart, we talked about how flow of ions across the membrane can result in a temporary change in membrane potential, which triggers the cellular response. G protein-coupled receptors, or GPCR, are a huge family of receptors. These receptors transmit extracellular signal through the activation of GTP-binding proteins, or G proteins. GPCRs are proteins that extend over the cell membrane 7 times. Because of this 7-helix receptors, or serpentine receptors. After ligand binding, a conformational change activates a resting heterotrimeric G protein. The G proteins that associate with GPCRs are a type made up of 3 subunits, like 3 blades of a fan. The alpha, beta, and gamma subunits. The activated G protein then interacts with either an ion channel or an enzyme in the membrane. Cell signaling using G protein-linked receptors occurs in a series or cycle. First, binding of a ligand to a GPCR. This causes exchange of GDP to GTP by the alpha subunit. Then the alpha subunit separates from the beta and gamma subunits, triggering a cellular response. Now the activated G protein has 2 parts, one consisting of the alpha subunit and the other beta and gamma subunits. The activated G-alpha binds with 80-nilyl cyclase and initiates steps to bring about cellular response, as shown here. The other path it can follow is that, after a while, the GTP on the active subunit of the G protein is hydrolyzed to GDP, and the beta, gamma subunits are deactivated. The subunits reassemble, forming the inactive G protein, and cycle starts again. Disorders associated with G protein-coupled receptors. Some disease-causing bacteria release toxins that interrupt G protein-coupled receptor signaling, leading to illnesses like pertussis, botulism, and cholera. Last thing that we will talk about now are the receptor tyrosine kinases. Receptor tyrosine kinases are a class of enzyme-linked receptors. A kinase is an enzyme that transfers phosphate groups to a protein or other target, and a receptor tyrosine kinase transfers phosphate groups, specifically to the amino acid tyrosine. First, signaling molecules bind to the extracellular domain of two nearby tyrosine kinase receptors. The two neighboring receptors then bond together. Phosphates are then attached to tyrosine on the intracellular domain of the receptors. The phosphorylated tyrosine can then transmit the signal to the next messenger within the cytoplasm. Receptor tyrosine kinases are essential in many signaling processes. For example, they bind to growth factors, molecules that promote cell division. And also they are part of the JAK, STAT pathway that we have discussed in our immunology video. So that is the end of our video. Now you know all about the cell signaling pathways. Stay tuned to sqadia.com to watch more such videos.